Hi everyone, my name is Sissy De Armas and my presentation is on climate change and its effects on temperature-dependent sex determination in loggerhead turtles. And I think it's really important to know about this topic and see how our ecosystems are being affected due to this increase in temperatures and how it can affect our species within them. Just to give you a little background on our outline, so we're going to be talking about how global warming is impacting these ratios, and it's important to see that the ratio of male to female turtles, how they mate, and it's bigger picture that if we don't watch over this, what may seem like a small detail, this special species of turtles could go extinct, causing even bigger impact on our ecosystems. Then we're going to talk about developmental cycles in loggerhead turtles. So we're going to piggyback off of increasing temperatures and talk about how temperature dependent sex determination and how it develops the stage is between embryonic development in a turtle after it's laid, and how it's crucial to the populations in our ecosystem. And then we're going to talk about how the study came all together, the bigger details within the study. So understanding the developmental stages and the processes, and in the end, how the study is aiming to understand primary sex ratios in context of warmer climates, and why this study is interesting and what the main ideas of it is. So what major key points we take away and why it's important. Here we'll be discussing the increasing temperatures in our ecosystems and why it's such a major issue to be aware of what's going on with this particular species of sea turtles, because not only does it pertain to these loggerhead turtles that we're specifically looking at, but it's turtles all around the world globally, due to the fact that climate change is not just affected in this certain area where our research is taking place, it's happening worldwide. And if we don't try to adjust or figure out what's going on, it could have serious effects on what the chain continues to look like. So right now we're trying to figure out what the balance between female to male ratios in turtles is and how we can find it. And if the scales of this ratio are tipped the wrong way, we could have serious repercussions such as extinction of this species. So what we're trying to do is global warming has fluctuating temperatures, which are leading to sex ratio alterations between male and females. And just the smallest tip in temperature could cause an overproduction of female or overproduction of males. Most likely it would be an overproduction of females due to the temperature for female production to thrive at 32 degrees Celsius and males to thrive at 25 degrees Celsius during their nesting period. So we'd be more likely to see the female population thrive during development, after they're laid to nest. And this is just one of the ways that temperature change could affect our turtle species. And it's really important to see how it may not seem like such a big deal since it's only like 2 degrees, 4 degrees Celsius increasing, but with animals that are temperature dependent for their determination of whether they will be female or male, it can make a major difference, and it will lead to an overpopulation of females. This overpopulation of females could lead to less males, leading to less reproduction, causing less eggs being laid, diminishing the turtle population, which is how they become extinct. So it's really important to see this, not just as people who are animal lovers, but worldwide, how turtles fit into our ecosystem and how it's all connected. If we don't have turtles, we don't have another species, and so on. The chain continues going. Another example I could pretty much think of is honeybees and how they're also becoming extinct. And it's just a chain effect of one issue to another to another. And if they don't pollinate flowers, we don't get, we can't farm and do all the special things we need to, which would cause major issues. And you can see how even a smaller insect is it plays such a big role in our ecosystem. So just something to think about when you're thinking about the future and how we survive in it and how all the other animals around us survive in it. They play a pretty big role, and not having them could cause serious issues for us. Seeing the developmental cycle and loggerhead turtles. So we're specifically trying to point out certain stages within their embryonic development, which leads to the female or male characteristics between the two. So first of all, we'll start off with diving deeper into the issue that we need to understand what causes these sex determining factors during embryonic development in these sea turtles. And here is an example of the stages, how they're being pictured from different stages in their life cycle and developing. And then we are talking about the thermosensitive period is where it all starts. And in this period, it's where the sex of the turtle is decided during development. So here, the gonads, which are the organs that produce gametes, which lead to the organs of testes or ovaries. These gonads are where the testes and ovaries are developed, depending on which temperature their nests are at, which could be fluctuating due to changing environments. Here in the gonads, there is a enzyme, which, if you didn't know, an enzyme is a protein which tries to accelerate a reaction. So this enzyme is converting sex steroids into ovaries or testes, called aromatase. So these aromatase levels are what influences the sex development of either females, which would be estrogen, or males, which would be androgens. So the lower the aromatase levels, the more likely for male characteristics being developed, and the higher the aromatase levels, the more likely for female characteristics to be developed. The male characteristics are usually around a lower temperature of 26 degrees. That's why it would say cold, because they prefer, when developing, it's more preferred during colder temperatures. And female characteristics would usually be developed around 32 degrees Celsius, hotter than the males. Not as hot as one would think, but increased temperatures lead to more likely female characteristics. And these factors are based on how the thermosensitive developmental stages work, but they can be hard to determine sometimes due to the turtle's lack of sexually dimorphic external characteristics. And these external characteristics are trying to specify on how they look. For example, a sexually dimorphic external characteristic in marine turtles would be their size. That's because externally they're different sizes. They can vary from male to female. Usually the male is larger. And these individuals are sexually dimorphic because they are individuals of different sex from the same species, and it can make it a lot harder for people to tell the difference between them due to the similarities. So they have a few sexually dimorphic characteristics. But their incubation period is critical to their development due to the conditions around them deciding whether or not their ratio will be even out in nature, uneven, there will be an overpopulation of some or not. It's critical to understand why naturally out in their own personal ecosystems when being laid, their environment is crucial to their development. It all depends on where the female wants to nest her eggs. Here we will be explaining the experimental study conducted by the scientific department of Earth, Ocean, and Atmospheric Science at Florida State University in 2017. This study was conducted by placing certain eggs in a condition determined by certain proxies, and these proxies are conditions that are controlled to try to explain why different proxies are affecting this. So pretty much how these changing incubation temperatures are determining the development of their testes or their ovaries while developing in their embryos. And so we can understand the dynamics and the structures of ecosystems and their environmental potential. So pretty much these proxies are non-invasive, causing the experiment to be non-invasive. So it is actually a field study instead of an experiment. But these correlations are based on things measured at the particular site. For example, some are measured completely during their whole incubation period. Some are placed at different sites where, due to the change in climate, it could change their temperature, changing their proxy. So we use these species with temperature dependent determination that depend on that, and we have their sex ratios altered due to the changes in temperature we have noticed. And overall, the goal was to collect data on the incubation period and the nest, affecting whether or not the conditions outside have altered the sex ratio between female and male turtles. To begin, we're going to talk about the hypothesis of this study, which it was estimated that the ratio of natural loggerhead turtles, and there you can see its scientific name, Caretta, and they're taking these turtles, they're taking approximately, I believe it was like 43 nests, and they're placing them divided evenly into 8 different proxies, which would change the conditions at which these developing turtles are growing at, and it will show you how the ratios differ from each proxy depending on its conditions. So we're trying to see how these developmenting factors around it are affecting the female to male ratio. And it's important to see up here in the picture, you can see they all start beginning after being nested, and from then it's decided whether it's going to be female or male. So during its development, crucial time period in between. So we begin the study with the nesting season of 2015 back to 2014. So here you can see the more precise date. There were 43 loggerhead turtle nests placed, and this took place in an area called Bahia, Brazil. So although we did take this data study from one area, it could potentially be for global studies to relate to due to it being on the coast. It's still an ocean with beaches and eggs where turtles lay their nests. So this is a very big issue worldwide for turtles. So these turtles were placed at 6 constant temperatures that range from 27 degrees Celsius to 30 degrees Celsius. And 5 of the nests were although removed from the analysis due to abnormal temperatures, most likely due to predation or consequential exposure since out in the wild. But we did get over half our data, so it was a success, essentially. With the remaining nests in the study, we continued by placing a handful of the nests in the proxies. So we divided up the 43 subtracted by the 5, the 38 nests left, into the 8 proxies. And these proxies were variables that used something immeasurable. So, for example, one of the proxies was sex determination only by temperature during the whole incubation period. So this helped single out which one and helped factor out those truly affected by sex ratio due to male and females based on certain temperatures in that area. Giving a better idea, there are 8 proxies, so they all differ, and if placed in these certain conditions, it would vary throughout, but that's sort of the gist of what we're trying to get at. Okay, so here are the 8 proxies of the experiment, and this is a good way of showing how we determined the ratio between turtles and their proxies. And since it was non-invasive and easily measured in the field, we used to estimate the ratio of marine turtle hatchlings. So in this graph, we have the 8 proxies to show how the loggerhead turtles were nested and observed and split up into these separate controlled environments. We're gonna see how it's based off of the first whole incubation period and the middle third incubation period. Those are the most crucial time periods throughout the study that scientists noticed were most affecting these turtles. So that's what they mainly emphasized looking at. And the temperature dependent period in this development era was helping show how the global change leads to this study of the proxies, because these are, although controlled, we're trying to alter a few things to see how it would change from the female ratio, what would raise it, what would lower it, and which we're trying to figure out which groups of turtles, depending on the proxies, were better produced under certain conditions. So which turtles, if turtles were better determined during the thermosensitive period between their ratio, or if temperature during thermosensitive period, just the middle third period, was the greater time to observe and record data. So this would be around the time where scientists would lay out the eggs and observe them throughout the time period recorded. In this table, we're trying to show the sex ratio estimates during the complete incubation. And it's trying to show how overall in sex ratio estimates among the whole incubation period, according to each individual in the proxies, the 5 whole period incubation proxies are more likely to produce female population within the female to male ratio. This goes for all the proxies due to the underlying factor that increased temperatures favors female development. So all the proxies with higher temperatures were more likely to produce female characteristics during development, leading into a higher ratio of females. Although the duration of the whole incubation does allow for a more precise and faster assessment, which includes the incubation conditions and the more specific development of the turtle, it can give incubation results showing how at higher temperatures there's females, and it can risk the idea that if temperatures were to increase, we would run into the upper thermal threshold, which are the limits at which the temperature hypothesized can reach, which could end up being bad for the females if temperatures rise too high. Although the females do thrive in the high temperatures, it could lead to the death of these because it can't be too hot for them. And it can trigger active responses such as death in the population, which would also be bad. So most importantly, we're trying to figure out which proxy would be the best to use. We have to consider both the reliability of the sex ratio estimate, and we have to think about the logistics of collecting the data on each side. So here you can see how when under the temperatures of the proxies, the female ratio was higher than the male ratio, saying how there were, under these proxies conditions, higher temperatures to help thrive the female ratio. But we have to think about how in all populated areas of loggerhead turtles may exemplify experimental error with our experiment, but without it, you can see how environmental conditions can affect it. This is why out of the 43 that we had, only 5 were due to experimental error. So we were able to keep over half of our data within the remaining 38, which is good for us. But now, naturally out in the wild, we can see that logistically there are many more errors that could factor in. But it would show a bigger, higher rate of natural results. So it's showing that the sand nests that varied from the 23 degrees to the 35 degrees in comparison were a lot higher to the female favoring ratio. And this was sort of a range a little outside of the temperatures that we estimated, but it showed a more natural results, like mentioned before, due to any environmental changes or fluctuations that could happen outside. So the embryonic growth rate shown within this study had around an estimate of 34.5 degrees Celsius, which proved to be 2.7 times higher than the normal testing temperatures in these loggerhead species. And if they were to rise anymore, it could have also serious ramifications on the female production. And in conclusion, we were able to figure out that there was a higher rate of female to male ratios due to higher increasing temperatures. This information was based on the temperature-dependent hatching success in natural conditions. And to be able to understand the climate change on these developmental stages is crucial and a very swaying factor for these species. Since these animals do rely on temperature for sex determination, which is the temperature-dependent sex determination factor, we need to realize how none of it can be constant since our world is so constantly changing, especially with climate change. In natural conditions, the temperatures would be seasonal, so that already affects the growth rate of the embryos, and it can be expected for it not to be constant. But since we have fluctuating temperatures, it's not more likely to be seen at a more constant range. If recorded throughout a whole time span of a year like the study was, you would see much more variation, which could affect the population of male to female turtles out into the world. That's why the thermosensitive period for the sex determination can be very depending on the environmental variations, because if in the summer where we have hotter sand temperatures and more likely to see female production, and then in the winter when it gets colder and we're more likely to see a little bit more male production, with these fluctuating temperatures it could change up the whole ratios, affecting overall what we have out into the oceans and causing a less reproduction of turtles overall. So with these increasing global temperatures, there's a higher probability of female development, putting the entire sea turtle species at risk, which could be a really big issue. further on for both us and the turtles and the ecosystem the turtles thrive in. And it's just really important to see how some things that we're doing can affect the world outside. And it's important to be conscious of our environment and to understand that we're just pretty much hurting ourselves in the process, let alone animals who are just living out there. So just something to keep in mind when you're not doing the right things for your environment or being a little wasteful in these situations. And I hope you guys enjoyed my talk. Here are my references, and this is why it's a little important to see how our environment is being affected, because you never know what's really happening out there in a big old world, just a little you never know what's happening. So if you have any questions, feel free to reach out to me, and thanks for listening to my talk.